Good morning everyone present here. It's myself Archana Patil from Patil Central School Halga, and it's a pleasure to present a case paper on STEM DLD over here. So what my topic which I have selected, the initiative which I have taken is DNA extraction from the different plant sources. And something which I have selected it, practical basis, hands-on STEM, hands-on practical basis. So to begin with DNA, this is the table of the contents which I'll be going through one by one. All the things will be covered. So introduction. So what exactly, just let me brief up what it is. Like if I tell you what is cell biology, right? If I tell you what a cell is, you all will tell it is a basic and fundamental unit of life. And I'll be telling you a structure of the cell. I'll be telling you I'll show you the video, I'll show you the pictures, everything will be shown to you, right? If I tell you, right, it is a cell which is circular, which is having its own membrane, cytoplasm is there, nucleus is there, nuclear membrane is there, DNA is there, this all becomes theoretical. Right, it becomes theoretical in the sense if I tell it is difficult to grasp what it is, because this is basically deals with the molecular biology, as DNA is a macromolecule which is present in the nucleus of the cell. So we have extracted that DNA and we are going to show it to the students. Actually the students have done their work, they have done it, extracted and showed it how exactly the DNA looks like. So this topic belongs to cell biology which we have taken on. So as I said, DNA is the genetic blueprint of life. So what was the need is, if I tell you theoretically like DNA looks like this or it looks like a thread-like structure, it becomes difficult. But what if I actually show you how it looks like, right? So that is what the need we have addressed. Because it is difficult to grasp the molecular biological concepts, we have done hands-on practice to show exactly this real life molecule, how it looks like to the students. So target group, what we are focused is, we are focused on class 10 or grade 10 students. They have actually involved in this and they have done the benchmark. Next. So what is DNA extraction? So what all it leads to, and how all different fields it is like. It leads to, or it is connected to medicine, forensic, genetic engineering, which is in detail we are going to, because it is a huge topic, genetic engineering, molecular biology. So base for that is DNA. And how it looks like, if you understand, then you can carry out the further analysis of DNA. So how my activity is associated with STEM, right? To begin with, so STEM science, as we all know, cell biology is a topic in the science subject for the 10th grade students. So that links to the science, obviously. Next. So this is all the introduction which has been shown, how the cell looks like, how the nucleus looks like. Next. Next. So technology, how it deals or relates to technology is, whatever DNA extracted, you have extracted, you can carry out a PCR technique, which is polymerase chain reaction. You can amplify, you can produce multiple copies of the DNA which you have extracted based on the sample size. If it is very less, you can go for PCR, then go for gel electrophoresis, wherein you are going to study DNA as separate bands and compare various DNAs which you can extract in the form of bands and analyze it. Finally, DNA sequencing, where you can go for nucleotide, that is base pairs. As we know, there are 4 base pairs: adenine, guanine, cytosine and thymine. So that can be done using a DNA sequencer. So this is how technology is related to our experiment. Next, engineering. So in this we have focused on the problem solving aspect. Like we are going to design a problem which has been created. We are going to design a solution for that. So this is how we are using the engineering principle. Suppose if the concentration is less, the sample size is less, how you are going to solve it? How you are going to solve it? So that is what we are involving engineering principle to solve the problem. Next. Mathematics, as I said, we will be using different reagents for doing these experiments like salt, detergent, alcohol. So in order to calculate their concentration, ratios and percentage, we will be using maths in the particular experiment or initiative what we will be doing. So objectives, what are the objectives we are focused on is, first we are going to demonstrate the DNA which is present. As we know that DNA is present in all living cells, so we are going to demonstrate the students that DNA is not only present in one sample, but we can extract it from various samples. For that we have taken different plant sources which I will be dealing in the next slides. So objective, what we are focused is practical exposure to the students. Then the scientific skills like observation, analysis were developed through this experiment. Finally the collaboration. So whole class of 10th grade I have taken. In total we have 37 students. So where we have made 4 groups. 4 groups, each group having 4 to 6 students. So all were involved in that and each one has done the benchmark, because I have taken 8 samples and distributed equally among all the 4 groups. So that was done. Finally they developed their critical thinking, that is observation and they were inquiring all the questions, and they came to know that different samples of DNA can give different yields of, different samples of plants can give different yields of DNA. Next. So this is the description of the practice, what we have followed. So we have used different plant samples like fruits, vegetables, flowers and fruits. Each two we have used. So for fruits I have used banana and orange. For vegetables, potato and tomato. For leaves, rio leaf and spinach leaf. And for hibiscus and marigold as the flowers. So from all these 8 samples we have extracted the DNA in our school. Next. So this is the benchmark what we have set up for the students, and all the samples were very local people which were available in the surroundings. They have brought the fruits from their home, and then it is the samples which were in our surrounding, in our garden we have taken, and we have done the extraction. Next. So procedure, it is a very simple procedure, just a 4-step procedure. So first what we have done is mashing. We have taken the sample, washed them thoroughly, then we have mashed them using a mortar and pestle with water as a solvent. After that, second step we have done filtration. What is filtration? Is we have filtered the extract so that we get the filtrate where actually our DNA is present. So after that filtration, we have taken reagents too. We have added 2 reagents, which is salt and detergent. So what these do now? What is the role of salt and detergent? I said you the structure of cell. It is having cell membrane, nuclear membrane, and DNA is present where? In the nucleus. It has to come out of the nuclear membrane and come in the cytoplasm, right? So for that you need to break the nuclear membrane. So this salt and detergent, what they do, they break the nuclear membrane and release the DNA into the cytoplasm of the cell. So it becomes easy for us to calculate or collect that DNA which is being come out of the nucleus. So final step, how the DNA we have collected separately is by precipitation method, where we have added ethanol, which is an alcohol. What it does, it clumps all the DNA molecules together in the cytoplasm and separates it from the cytoplasmic contents. So that we can extract, we can separate using other techniques, and we can go for technological aspects of the STEM. So methodology, yes, pedagogical approach where students were inquiry-based approach, it was an experiential learning. The role of the teacher, just a demonstrator and facilitator. Next, role of the students, they were active experimenters, where they were observers, they have kept the records, they have done it by themselves. Curriculum integration, as I told, it is one of the topic in the biology, that is cell biology. Next. So methodology, these are the method what we have followed. Next. Preparation, first our students have done the bench work, they have arranged the samples, they have done themselves crushed, mashed the samples, and then they have extracted the DNA. Next. Filtration, which is done using a simple filter paper in the laboratory itself, and we got the filtrate of different samples. So one I have shown you, like that we have done 8 samples total together. Next. Reagents, as I told, next. 2 reagents we have used, just a simple liquid soap or detergent which is available in your homes, in your localities, in your kitchens, we can use that, plus salt. So these 2 reagents we have used, which are easily available in your hand. So there is no need as prior setup to do it for 1 day prior. Instant, in 10 minutes you can set it up and go for extraction of DNA. Next. Next. Finally, the precipitation, as I said that it appears. DNA, as you can see that different layers, 2 layers have been formed, wherein the above layer, top layer, which is one phase, is nothing but your DNA which is extracted. So all the samples of fruits, vegetables, flowers and fruits have displayed it in the test tubes, which our students have worked with. And then we have extracted the DNA, which forms a separate layer on the top of the test tubes. This is the observation that DNA looks thread-like, cloudy, floating at the interface between the 2 liquids. Next. So this is what about the role of a teacher and a student. The student was a hands-on experimentation they had learned. Then it was a team collaboration, critical thinking was developed, then the presentation and reflection that they have understood the concept of genetics thoroughly. The role of the teacher, first I had introduced them, I demonstrated them the experiment once, then later I have assigned 4 groups to do all the samples individually. Next. So evidence-based impact. So what was the evidence-based impact? First is student learning outcome. What they saw, they never thought they can see the DNA with their naked eyes. They never thought it. Unless we were just telling them that DNA is present, where it is present exactly. We say in the nucleus. But how it looks in the nucleus? Has anyone thought of this? No. But we are supposed to show them how the DNA looks like. So this is what we can see with naked eyes without using any instrument, without using a simple microscope also. So that can be shown, and that made the concept of molecular biology very, very clear to the students.
Ma'am, just one question.
Yes.
How did you confirm that is DNA?
We need to go for further. That is what culture. So because it appears thread-like structure.
Basically, because I am the chemistry person.
Yeah.
Okay.
Yes, sir, yes.
So just I am interested in that. You extract the things from the plants. Apart from DNA, there are many other chemical compounds which will come along with the DNA.
It will come along with the DNA. For that, I said, sir, sorry to disturb, we will have to use the technological aspect. I said PCR, so that is one gel electrophoresis. They are specifically meant to only show the DNA.
Okay, I just want you to define PCR.
PCR is a polymerase chain reaction machine.
Explain it.
So what it does is, suppose you give DNA to it, it separates the two strands of DNA, right? And then we have to add nucleotide sequences, primers to it. When we add that, it goes on producing multiple copies of the same strand of DNA. So that whatever DNA was just two-stranded, you can get n number of copies of it. So that is what a PCR machine does.
So at what temperature you have done extension of the DNA?
So we have not done this work, ma'am. PCR
so how
machine
did you
is
get
not
DNA?
DNA is what we have extracted using the solvents.
आप बोले जा रहे हो वो न्यूक्लियस में isn't it in the chromosomes?
In the chromosomes.
Yeah, it is
in
not
the
one organelle which will come out of the nucleus.
Yes, ma'am.
It is in the chromosome. So please बोलिए कि वो chromosome में से आपने extract किया. आप कह रहे हो nuclear membrane टूटेगा, chromosome DNA निकल आएगा. ऐसा नहीं होता है.
Okay.
And please see, your study seems very theoretical. I'll tell you in a very, very, honest way. It seems very theoretical. उसमें बहुत सारे जो हैं चीजें, अगर उसके बारे में questions पूछा जाए, आप explain नहीं कर पाओगे. This is like
just
a lot of.
we have
it is a lot of it is not done only actually. लेकिन when you present something like this, you must have a proper. आप कह रहे हो ना Vim और salt से DNA extract हो जाएगा, ऐसा नहीं होगा.
It is a plant-based product, man. Plant-based we can do it.
Product, but please read about it.
Like plant-based products can be done easily, ma'am. What we can do, animal-based product, if you are going for any cell line structure or that, we cannot do it with these simple reagents, ma'am. We need to use the buffers.
Simple reagents in only one stage of it. It's not that you put Vim and salt and DNA निकलेगा. Like what sir is telling, there are so many components in a cell. It's not only with that you will get it. Please take this as a suggestion. This is not a criticism.
Sure, ma'am.
Okay? Sure. Yeah.
No, my question was when you see basically you have taken water as a solvent for grinding that. So all the components will come into water.
Yeah.
You are going to add ethanol as a precipitating agent. Okay. So when you add ethanol, so both water and ethanol are miscible. So when both are miscible, whatever the third component will come out definitely.
Yeah.
And that third component may be n number of compounds which are present in the plant.
That is what, sir. We are going to show them. This is where we can extract. But that further study has to be carried out with different machines.
So in that case, what I mean is just suggestion. What I mean to tell is you cannot 100% tell that it is whatever extract we have got. It
pure
is
DNA. I'm not telling it is a pure DNA. It is just a process what we are showing. Pure DNA cannot be done by these simple techniques, sir.
And until you centrifuge, how are you going to separate components?
That we need to take it and centrifuge later, ma'am. Centrifugation upper layer I'll be connecting. That is the end. As per your, I got it. We can do it further very analysis like using PCR, gel electrophoresis. What I said. This is just a simple technique which I have told them. This is how we can extract the initial phase of DNA.
Okay.
Further, we need to study it.
The logical end to that would be probably whatever you have extracted. Like you said, it is the initial thing. Probably if resources were available.
Yeah.
Just authentic. Yeah.
That is what in technology I told, ma'am. PCR, gel electrophoresis. This can be carried out further for the sample what has been extracted. So knowledge gained was the DNA is isolated by simple techniques. Next testimony is what students say the science has become real today because they could see something new out of extraction.
But I really beg to differ here. We have isolated DNA when I did my MSc from University of Pune in 1993. So it is not as if DNA अब लोग देखने लगे हैं. DNA for decades and decades
But
देखा है.
at school level, ma'am.
Yes.
At school level.
तो आपने ये बिना gel electrophoresis के बिना
I have done even my PG have completed in gel electrophoresis PCR. My work is done.
So were you able to show it to children?
Yeah, we have done, ma'am. Whatever my PG work, that presentation or data, what we have documentation, I have done in that only, and we have to do it.
Then that is a good thing if you.
So I have initiated this to just demonstrate them. So because my work is done, because unless I do that submit, I would not get my PG. I have done on that only.
No, we are talking about what you did with children, not.
Yeah, with children, this was just initiative which I have taken up.
Perfect. Chaliye.
student giving a simple worksheet, like simple questions were asked and they have solved it and they have displayed it. I have displayed board which is the worksheet which is solved by the student. Simple questions like what is DNA, function of DNA were asked, and what is the role of salt detergents, everything was asked and they have answered it properly. Next. So design of this, next. We have covered all these cognitive domain, Bloom's taxonomy, like how they understood it. That's simple to just demonstrate. I have given the slides. Next. Affective domain, wherein student they were curious about lab practices, what we done. Next. In psychomotor domain, practical applications, like they went to the lab, they did it personally. That was all the psychomotor domain which was covered. Next. Novelty. So what was the novelty? Might be I told you we have extracted DNA. So what newness I have done is not from single sample, but I have given them 8 total samples where they can easily compare what amount of DNA can be extracted from each different sample. Like maybe in some samples they gave it low, especially the orange and marigold flowers which I have taken, they gave very low concentration of DNA. Other they have given which could be visible to us. That quantity or concentration of DNA was available on the slides. Next. So this is the unique aspects of the practice, what we have done. Integration. Integration, multiple domain learning, then visual aspects were focused on. This was perfectly student-centered activity because students have done it, experiential learning was carried out. The next, it promotes scientific temper and a research methodology among the students. At least some basic work they are doing and they try to understand what exactly, how the research basic step is. That can be focused on. So included, what is included next? Included in this particular set, each and every student of the class was included, let him be a slow learner or a topper of the class. All the students were, the work was distributed equally among them. So each one of them have done the work and they have understood because it was no single student left out in the class. So materials also, they were very locally available. Each one had brought the fruits, vegetables from their home and the flowers and leaves from our garden only we have taken. Next, scalability and replicability. Yes, this can be done in any school because it is very simple concept. All the materials will be easily available. And then necessary condition, just a basic simple lab setup with all the instruments. Simple instrument like glass, test tube beakers can be done. Next is documentation and step by step what slides or 4 methods I have shown you. That can be followed. Sustainability and cost effectiveness. So what is sustainability? How long it can be done? As long as living materials are available, living things are available, we can go on extracting the DNA from each. So that is long-term viability of the initiative. Then budget, almost less than 200 rupees was the budget because it's very easily available materials and all the local fruits and vegetables only were used. Next. So learning outcomes. So what we learned is orange and marigold, they produce very less DNA as I told you. So it pre-chilled ethanol. We kept the ethanol in the fridge, not directly the normal ethanol temperature, and then it gave us results faster. Next. These are the key insights which the students have taken. Finally, to conclude with, so what we can see is DNA just can be seen, just cannot be studied. And then it enhances curiosity and teamwork, inquiry-based skills among the students. Future, we can even show them using the plant cell, apart from plant cell, animal cells also, and we can demonstrate the gel electrophoresis technique. So simple quote I would end with, seeing the invisible actually inspires the future. So this is what I would like to give my quote from my end. So whatever invisible things, we can see them and that will inspire the future generations. Next. So this is my team, 10 standard actually base pillars of the work who have done it, bench work. So these were the students who have actually done involved being in the groups and they have extracted it and that was the results what they have presented. So this is the QR code, one video I have uploaded. You can scan this QR code please and go through the video which our student has done the work and shown it.